All right, well, it's right about 2:30 Eastern time, so I wanted to go ahead and get started. So thanks so much for joining us this afternoon here at the ASGH Virtual Meeting 2021. My name is David Higgins, and I'm here today on behalf of the Gabriella Miller Kids First Data Resource Center. And so what I'll be presenting today is our Kids First information session. This is just a broad overview of what Kids First is, what types of data sets the Kids First DRC offers, and how we can support your research in the study of human genetics. And so I know normally when we love to attend ASGH in person, and we've been there for many, many years. And so we are still here at ASGH, although ASGH is, of course, all around the world now. And so when we're back in person next year in 2022, we can't wait to have you again at our booth for a coffee hour and poster session to talk all things Kids First in person. But here today for the virtual meeting, what I'll be providing is a bit of an overview for who we are at the Kids First DRC and what types of services we offer. And then I'll introduce some of our different platforms that can support your research into human genetics, specifically looking at pediatric cancers and birth defects. We'll have some links at the end for getting started, including how to apply for access as well. And so I introduced myself at the top. So my name is David Higgins. I'm a program manager here at the DRC. I'm employed by the Children's Hospital of Philadelphia. And my role here at the DRC is very much involved in user outreach and training. Serving as the front-facing person who can talk to all of our exciting research collaborators, such as yourselves who are joined here today with us, and really kind of trying to help everybody out to be able to complete their research and connect to the rich set of resources that we have here at the DRC, which hopefully we're going to get into here today. So before I dive into the content, I just wanted to share a little bit of housekeeping. That our session is being recorded today, and we may be uploading this to our YouTube channel in the future, so that way we can provide it for folks who couldn't attend the live session here this afternoon. We do encourage everyone to participate, so the chat feature is active and you can post in the chat. I've got a couple assistants here, other coworkers here at the DRC who will be helping to monitor the chat and help answer questions. And I'll be able to hopefully have some question time here at the end as well to be able to respond. So please be active in the chat and feel free to post any questions that you have there, and we'll be able to address them throughout the talk. All right, so maybe we can get started with what exactly is the Kids First Data Resource Center. So our goal, so what are we? So we are a collaborative pediatric research effort with the goal of understanding the genetic causes and links between childhood cancer and structural birth defects. So that's our kind of our mission statement. And so to pick apart different pieces of that, we are a collaborative effort. And so the Kids First DRC understands that human genetics is incredibly complicated, and especially each of these different cancer types and birth defects are genetically complicated. And so no group can identify everything on their own. And so we understand the power of collaboration and supporting researchers around the world and basically getting as many smart people as we can to look at these data sets and do their research with our data sets can support real discoveries that can in turn support children, which is our goal. And so as part of our goal, we have these kind of four main aims. And so we have developed these data-driven platforms that allow users to be able to access these very large genome scale data sets and clinical data sets from our different disease types. Again, we are empowering collaborative discovery and partnerships between different groups, so in order to be able to support cross-disease research. Ultimately, what we want to be able to empower then is rapid translation to personalized treatment. So taking this information out of the genome and into the clinic, where clinicians can then begin to use our discoveries that we make with the Kids First data into ultimately helping children, which is of course our true goal. And then finally, doing that as quickly as possible by accelerating the discovery by pooling all this large data set together. And so if you haven't been to a Kids First talk before, then you might be wondering, well, why are we studying pediatric cancer and structural birth defects together? And so a paper came out in 2019 that was published in the JAMA Oncology Journal, which basically looked at 10 million live births and compared incidences of pediatric cancer and structural birth defects. And the results, to sum it up in one sentence, is that children born with birth defects have an increased risk to develop cancer. And so what they suggest to the authors and suggest to us is that there's a common underlying cause between structural birth defects and these types of cancers. So therefore, we should study them in tandem in order to be able to understand how these different pathways might be interacting with each other inside our patients. So Kids First has been around for a long time now. I said we've been coming to ASGH for many years, and so we've been at it for quite a bit, and now we've released up to 27 different data sets are available to apply through dbGaP for any researcher to be able to use for their research projects. And so these are our release data sets, and so I'll allow you to be able to kind of peruse them. We've got our structural birth defect studies on the left-hand side, our pediatric cancer studies on the right-hand side, as well as what I call our combined studies, which actually, even within that one study, we're looking at both cancers and birth defects. And so our big overall summary stats there is we have over 76,000 files from 18,000 participants, and we've just hit 1 petabyte of data. So we have so much information and so many files and genetics and genomes that we want to share. We want to support research, and we want to get more people using them, looking at them, applying to research them, and getting more people involved with Kids First and the Kids First DRC. So that's a lot, but we're not done. So we also are funded for many additional data sets will be coming soon. We just awarded the 2021 XO1s were provided by the Kids First program. And so here's a preview of those, which will be coming soon. So those, as far as those files, probably won't be available for at least 12 months from today. But you can go on the NIH Kids First program website and read about the full list of Kids First supported projects to see our full set of data. So what do we do as the Kids First DRC? So we support these different projects by harmonizing these data sets from all of these different researchers, from all of these different groups, and then we harmonize them together to allow this cross comparison. And so one thing that we do is we harmonize the clinical data. So we receive clinical data corresponding to each of these patients and participants that's been enrolled in these Kids First studies. And we harmonize them so that way we can compare the diagnoses and the phenotypes from the different researchers, which will allow us to be able to compare across. And so as far as our clinical data, we have, it's harmonized to 2 different ontologies. And so we're harmonized, our phenotypes are harmonized to HPO, as well as our diseases, which are harmonized to the Mondo ontology. And so assigning these specific diagnoses to these specific ontologies then allows users very rapidly to be able to search across the different data sets, using this one standardized set of terms. And so here's a model. So the HPO ontology is hierarchical. And so there has a kind of a nesting set of terms, which is we'll go into a little bit later today when we explore the Kids First portal and see that live in person. So here's our different HPO terms with their associated HPO numbers. So Kids First is also genome data as well. And so our genomic data is also harmonized such that the genomes from the Kids First neuroblastoma study can be directly compared to the genomes from the Kids First heart defects study. And so how we do that is that we run our genomic data through a consistent set of bioinformatic pipelines, which our team of bioinformaticians has developed to be consistent with the GATK Best Practices. And so we provide a couple different sets of genomic data depending on the individual projects. It might be different sets might be available, but we almost generally have germline variants available, especially for the proband participants. We also run a trio-based joint calling pipeline. So for many of our studies, we receive 3 sets of DNA. So we receive a DNA sample from the participant himself. the child who has the associated diagnosis, as well as we receive a DNA sample from the child's parents if it's available for that particular study. So those three individuals, the two parents and the one child, are joint called together. And we use this to allow us to be able to identify how particular alleles might be inherited between the two parents. And it also allows us to be able to identify de novo mutations that might be spontaneously arising in the child, which might be resulting in these particular diagnoses or phenotypes. So those are the germline variants. We also have somatic variants for our cancer studies. So these are the tumor normal comparisons. And so when a tumor has been, if a tumor tissue is available from the particular patient, we sequence the genome of the tumor as well. So we have that genome available, and then we run our different somatic variant calling pipelines. And so that includes identifying simple nucleotide variants. So those are individual base pairs where a T turns into an A. We've also have called our copy number variants. We're looking at regions of the genome. Our whole genes might be duplicated, as well as our somatic variant pipeline. We're looking at large-scale chromosomal insertions, deletions, and those sorts of large-scale somatic variants. Some Kids First studies also have RNA-seq data available as well. So that is available in its raw form as FASTQ files, but we've also run our RNA-seq pipeline on those data sets as well. And we have generated our harmonized data, which includes gene expression information. So just a long list of genes and then expression values associated with them, as well as gene fusion calls called by our gene fusion pipeline. And so all of this perhaps sounds like a lot of all of our different software tools. And it is a bit of different tools that we provide, for these sets of files that we run through these different callers and aligners and sorting bioinformatic pipelines. But all of these pipelines are available, and so we provide information on the pipelines in our GitHub repo. And so if you're interested in seeing how we do it, and then you can look at our pipeline. If you want to run your own data through our pipeline, you can download it and run your own files as well. So it allows users to be able to try to be very transparent to all of our users that this is what we're using. And if you're interested in using it too, then we'd love to share it with you. So these are our standard workflows and deliverables, is these highlighted in green here. So how do we access Kids First data? So Kids First data can be accessed on our two different cloud platforms that we've developed here at the DRC. The first of which is the Kids First portal. So the Kids First portal is kind of the front door, so to speak. This is where users can get started with Kids First. You can explore our Kids First data. You can build these cohorts across the different studies, and you can explore different files that you might be interested in for your analysis. And then once you have those files, then you can move them over to our second cloud platform that we support, which is called GATK. GATK is our online cloud-based analysis platform. So this allows you not just to be able to look at the files and share files with collaborators, but also run workflows on your data in the cloud. So you don't have to necessarily do a large data transfer, to a local server. You can do the analysis all in the cloud through your browser or through a command line if you prefer. So that's what I'll be spending a little bit of the rest of our time here today, covering our two different platforms and introducing the different features of the portal and GATK. So a Kids First Data Resource Portal, I'll give a brief preview here in the slides. And so this can be accessed today. So anyone can log on to the Kids First Data Resource Portal. It's free to create an account. It's free to click around, and it's free to look at the different data sets in order to be able to do your analysis. And so here's the Explore Data tab, which again, we'll take a closer look at here in a second. So this provides kind of a brief overview of the different study information, different diagnoses, and different phenotypes that are associated with the particular participants that we are investigating. And then once you identify those sets of participants, then I'll show you how to find them, the files associated with them here in the file repository. And so this is where you can find those individual, BCF files or alignments, which you can then, download or push to GATK for your analysis. All right. And so maybe I'll show a couple more slides before I hop over to the live demo. And so once you have your files identified, there's this Analyze in Chromatica button. So you can select that, these are the 3 files that I'm interested in. And then, yes, I'm ready to analyze them in Chromatica. And so kind of demonstrating how our 2 platforms interact here is that we suggest users start here, at the Kids First portal. And from there, you can identify the different data you're interested in working with. You can also upload your own data as well. So if you have your own set of neuroblastoma samples and you want to compare them to the Kids First neuroblastoma in order to be able to grow your sample size, we can support that definitely. And so users can also bring in their own data and combine together to form a larger study to better support your research. From that point, you can enter into Chromatica, which is our cloud-based computing environment. We have different workflows, so you can select from our existing workflows, or you can bring in your own tools that you might have for your particular research, and then run those large-scale workflows on your data. And then finally, you can run it through what we call the data cruncher. So the data cruncher is what allows you to be able to explore those data sets, look inside those big text files. And so it supports Jupyter notebooks for R-based analysis, for Python-based analysis, as well as RStudio for R-based analysis. And so just a little preview of what Chromatica looks like. And so again, this is where you're able to run these large workflows. So this has the Rabix composer, and it looks like we are building the RNA-seq alignment tool here. And so you're able to plug and play these different pieces of software, have the outputs connect into different tools, and then you can ultimately generate the output that you need for your particular analysis. And then finally, this is a sneak preview of the data cruncher. And so the data cruncher allows you to be able to access those files that you have created. So you can select a Python notebook, you can select an RStudio, or you can even just select a terminal environment, which is what's shown here in this live demo. And so here we've got SAM tools running, and we can do SAM tools view, and it pulls up our view there of our alignment. And so this one is just kind of a preview of the different features that are available on Kids First, the Kids First portal, as well as Chromatica. Okay, so at this point, I'm going to take a break from my slides, and so we're going to dive into the Kids First portal itself, and I'll show you some of the features that we have. So maybe I can take a pause. So I see that some people have asked some questions here in the chat, so maybe I can go ahead and join in. So one person has asked, do you provide joint call data files with all individuals included in the VCF file? So unfortunately, no, we don't provide that. And so we do provide individual VCFs, so that way you have the individual participants that you're interested in studying. And we have the joint call VCF on the family level, so just the 3 individuals. So we don't provide the joint call across all individuals in the cohort because we want to allow users the flexibility to be able to identify their own cohort. And we also want to support users who might identify some individuals in this study and some individuals in that other study, and perhaps some individuals of your own that you can then run your own joint call on. So we support joint calling in Chromatica, and we have a tool that we allow users to be able to run, but we don't provide those files necessarily on our own. So that's the first question. So the second question is asking, how much does Chromatica cost? So Chromatica is a tool that allows users to be able to store files as well as run these analyses. And so I'll talk a little bit about cost a little bit later, but if you are storing the Kids First data resource files, and there is no charge to use it. If you are wanting to download those files directly, if you have your own high performance cluster at your institution that you're looking to work on, there's no egress charge associated with that. And so that is completely free. If you want to use Chromatica as an analysis platform, there are fees. So Chromatica is powered by AWS. And so when you want to do a large computing job, what is happening underneath the hood, so to speak, is that you're developing calling up these large-scale computers which are running the compute. So individual tasks can, of course, depend on whether you're running a very small task. So if you're running a SAM tools index generation, that might cost maybe 3 cents. Whereas if you're running a whole, genome alignment and variant calling, that might cost perhaps $2 per sample. So individual workflows will have different costs associated with, the scale and time that's associated with running those particular workflows. And then the data cruncher tool, again, you're operating on these powerful computers in the cloud. So the data cruncher bills on a per hour basis. And I think it runs, depending again on how powerful the computers are that you're operating, anywhere from about $1 an hour to maybe $2 an hour to be able to use those resources. So that was a question about how much does GATK cost. So I want to make sure I agree with that. Okay, so let me go ahead and dive into our live demo. And so I can go now to the Kids First portal. And so when you first log on to our website, so our website is portal.kidsfirstdrc.org/ that will take you to this page right here. And so you'll be prompted with a couple different ways to log in. And so anyone with a Google authenticated login can be able to sign in and create an account. And so once you do, so we'll ask you to accept our terms. So basically you're going to use our data for good, for research, and you're not going to try to identify the individuals associated with it, and you're going to use NIH best practices for controlled access data. And once you agree to those terms, then it takes you into the dashboard. So the dashboard is the big overview of the different data sets that are available. So within the dashboard, we have these different settings up at the top. And so I'll take us into the studies page first. And so the studies page, what this does is this outlines all the different studies that are available on Kids First. And so we have our different Kids First data sets here, which are outlined, but then you can narrow these down a bit further. So if I'm just interested in the cancer studies, then I could select cancer. And now we're just looking at the 9 Kids First cancer studies. So this gives you a bit of overview information. So it tells you the number of participants that are associated with each of these studies, and then the different data files that are available as well. So if we wanted to start from this point and perhaps look at the neuroblastoma study, then I can click on Kids First NBL. So this takes us over now. So now we've left the studies tab, and now we're in the explore data tab. So this is what I was alluding to in my demo slides there. So now we're looking at the exploring the data live in front of us. And so within the Kids First Explore Data tool, we can see that we've got, we're looking at participants from this one study, the neuroblastoma study. We don't necessarily have any phenotypes associated with this study, but we do have our diagnoses here. And you can see, of course, the most common one is neuroblastoma. We have demographic information as well as the age of diagnosis of these different participants. So if I wanted to start a new query, and so for this particular study, there's 1,600 participants and about 18,000 files. So if I wanted to start a new query, let's form a new query here, and you can see all my my math has readjusted here, and now I'm looking at the full set of participants. Let's say I was interested in perhaps heart defects. And so if I can spell heart defects, then what we can see is that there's a study, Kids First Congenital Heart Defects. So maybe I want to apply that. I want to look at all of the participants in the heart defect study. And so the heart defect study is one of our largest studies, and also has a lot of very rich observed phenotype data that allows users to be able to look at different phenotypes in these different participants in the heart defect study. And again, we've got diagnosis information as well, which is associated with these different participants. And you can add different subsets. So let's say if you're only interested in, if you study tetralogy of Fallot, which is a particular heart defect, and you only want to look at those participants, then we can narrow these 2,000 participants down. You can either click right here in the diagnosis tab, And now what we've done is we've added a filter. So now we're looking at the heart defect study and we're looking at tetralogy of Fallot. So now we've narrowed our search down to 142 participants. So we're kind of filtering out to just that subset that we're very interested in. If we wanted to look even further, so maybe we can look at particular observed phenotypes. And so I like to do that by going into the clinical tab and then looking at the observed phenotype button, which brings up our HPO browser. And so I talked a bit about HPO at the top, which is this hierarchical system of ontology terms that allows us to be able to describe different phenotypes of individuals. And so if we're looking at particular abnormalities of the cardiovascular system, we can explore and go down this tree in order to be able to identify very specific abnormalities, very specific diagnoses, and very specific phenotypes of these participants. And so if we specifically are interested in the development of the right ventricle, then we can just select those particular participants and apply that term. And so now we're down to 94 participants, and we have 340 files associated with those. So you can see we're kind of narrowing it down to be very, very specific and look at files for particular participants that we might be interested in adding to our study, our investigation. So at this point, you can go a couple different places. So we also have support a table view, which allows users to be able to look at these different participants in a large table instead of in the summary graphic view. You can also download, so you can download a clinical data manifest, which outlines all of those different participants with their phenotypic abnormalities in a big Excel file. So that's available as well under the download button. But for now, I'm going to look at these 340 files that are associated with these participants, and let's see what's available. And so when I clicked that button, what it did, it sent me from studies over to the file repository tool. And so this is now where we can begin to look at the particular files themselves in order to be able to figure out which ones are the ones that we might be most interested in. And so there's 340 files from 94 participants, and we have about 6 terabytes of data to explore here. So if I wanted to add specific file filters, so similar to the Explore Data tool, I can add filters, which will again narrow this a little bit further. So if I know that I just want to look at the individual variants, which we provide in GBCF format, I can click this GBCF button. And what this does is this filters us to just looking at the GBCFs for these participants. And so we've got 94 files from 94 participants. We're getting 1 file from each participant. So this is the germline calls. If we wanted to look at the variant calls, we could click the variant calls button. And so this takes us over. These are those trio-based variant that I described earlier. And this can provide us, these are provided in BCF format. Or if you want to run your own analysis, so if you want to start from the beginning, we provide alignments as well. For this particular study, it looks like we provide both either BAM or CRAM format. And so if you're interested in starting from the beginning, you can begin with the alignment as well, and these are available. So I might choose, for the sake of my demonstration here, let's say I'm interested in the GBCFs. And for the sake of example, I'm going to choose these top 1, 2, 3 files and say these are the ones that I'm most interested in for my particular analysis. So at this point, what I can do is I can take these files, and there is a download button. So if you are interested, you can download them to your computer, or you can download them to a local cluster as well, or we can analyze in GATK, which is this button right here. And so what this does when I click this button, this is going to push us over and it's going to tell me which project do you want to copy this to. And so I created a demo project here earlier today, ASGH demo. And so that's where I want to copy the files to. So I'm going to put it right there and click copy 3 files. Success, the files were copied to your GATK project ASGH demo. So we can click this link to open it in GATK. But I'm going to come in the front door there instead of clicking that link directly. Okay, so at this point, I'm going to switch now. So I'm going to move websites. So I'm going to move from the Kids First portal, and I'm going to move to our second website, which is GATK. So while I'm getting logged in here, I see there's another question. If we have credits on AWS, can we use the credits on GATK? And so that's a great question. And so I believe at this time that is not supported, but I can get you in touch with our support group, especially the Seven Bridges support, who would be able to explore that and maybe able to give you some more information about having specific AWS credits for your analysis. So when I go to GATK, so this is our second website, this is gatka.sbgenomics.com. So GATK is developed in partnership with Seven Bridges Genomics. And so they have supported us by helping to build our website here. So you can either log in with a username or password, or if you have an NIH ERA Commons ID, you can log in with that as well. We recommend logging in with an ERA Commons ID if you have it, because if you're authenticated through dbGaP to access different Kids First data, then that authentication will carry over, from dbGaP into the Kids First portal, into GATK, and it's, like showing instead of showing your ID, 3 different times, it's all going to be you're consistent throughout the full data set there. So I'm going to log in with my ERA Commons ID and log in. And I'm going to authorize that. And that takes me to the homepage. And so on the homepage, so GATK is organized into these groups of or areas called projects. And so GATK is set up to, like I said, you can store files here. You can move files from the Kids First portal over to GATK. You can store them in these different projects or different rooms, different areas, just like projects in a research lab with a wet bench. These are digital, virtual projects for doing genomics in the cloud. And so I'm, like I said, I'm involved in a lot of different training and outreach. So I'm involved in a lot of different projects. And so you can collaborate with different users and different sets of people on different projects. So it allows you to be able to control who has access to what files and invite other people very easily. And then on the other side, we have the analyses. So these are the work that users are doing in these projects that I have access to. So you can do more than just store the files. You can also run analysis as well. So I have my ASGH demo project up here that I created this afternoon. So I'm going to click on that. And so when you join a project for the first time, it provides you with this landing space. It gives you a whiteboard here that you can edit, that you can write notes. You can have different notes for your analysis. You can add a description here under the description box. You can also add, so I was saying you can add members. And so right now I'm all by myself because I just created this project on my own. But if I wanted to add one of our users, so maybe our bioinformatician, Jan Kuhn, I could add his username and then invite him to join. And then he would pop up here next to me. And he'd be able to see these same files that I'm seeing. So it's very easy to add collaborators within an institution, but it's also very easy to add collaborators across institutions for large-scale collaborations as well. So it makes that facilitation of those inter-organizational collaborations very, very smooth. So this is the dashboard, and then I'll show you my files here. So you'll remember I pushed over those 3 GVCF files, and so here they are in my GATK project. And so if I wanted to, again, I could then begin to work on them for my analysis. So by selecting them, again, there are ways to download the files. You can download the links. I can also show you we have some scripts that are available that allow you to be able to download Kids First data to a cluster as well. And so we definitely support folks working wherever they want to do their work. But I'll show you that GATK is one place that you can do your work. So you don't have to do these large scale transfers and downloads and things like that. And so this is a GVCF file. And so we can click on it, but I believe it's just going to be a large compressed data file. And so this is not very useful to do any analysis on by itself. I can't really look at it and get any useful information out of that. So where do we go next? So from that Files tab, we can go to the next tab over, which is the apps. And so at this point, you can either create your own app. So this is the Rabix Composer will open up, which is what allows you to be able to build your own tool or build your own workflow. So GATK supports tools written in the Common Workflow Language, CWL. We also support loading Docker images. So if your tool is Dockerized, it's very easy to upload the Docker image, and then you can immediately have it available here in the cloud. So it supports users who have their own tools and have their own apps. It's easy to bring tools into GATK. But we also have our apps, kind of like the app store on your phone. But these are free to use, or at least free to browse and explore. And so if I have I have those GVC apps, and so if I want to run maybe a joint call of those GVC apps, then we have a couple different options as far as the previously created ones that are available to run joint calls. And so we've got a couple. So we've got some GATK Broad pipelines. We have some Genesis pipelines as well. And then, excuse me, down here, we have the Kids First DRC joint genotyping workflow. So this is that tool that we've developed, and I demonstrated the image on the front of our talk, which shows the different pipelines that we provide. So let's look at this one a little bit further. And so here we've got that diagram showing the different steps that we provide for running the app on either your data that you're bringing into the cloud or running it on the Kids First data that you're interested in investigating. We've got a great README, which is written by our bioinformaticians, which provides information on all the different callers, all the different settings, and provides suggested references and inputs that you need to make the software tool run, as well as the graphical representation of what the workflow is doing. And then there was a question earlier about costs. And so for our Kids First pipelines, we've run these many, many times on all of the Kids First data sets. So we're beginning to get a sense of about how much they cost. And so, for these particular inputs that we've run, we find that they take anywhere from an hour and a half to 2 hours and cost between $2 to $3 per sample. So at least for the Kids First apps, we do provide some estimates for those costs here as well. So if I want to run this, then I can go back to that search page and I can click, this is the one I want to run. And what that will do is create a new task for me. And so this is now one instance of running that workflow. So I want to take all that, my 3 files that I identified on the Kids First portal and run them through this workflow. So because this is the first time I'm pulling this app in, it suggests that, hey, do you want to use all of these reference files that we need to run the workflow? So it has our reference genome, our different additional controls that we use in order to be able to make the software run correctly. And so I'm going to say, yes, thank you for pulling all that for me. And it's going to copy that over, and it's going to fill in these lines here. So at this point, we can then upload, you can then bring your own data. So if I want to input the VCFs, then I can select those VCFs that I copied from the portal and then save that selection, and now they appear right here. We can adjust different apps, different settings. So just like you're working on the command line, you can add different flags. You can adjust different settings for the callers to get it to be very specific to how you choose to use it. And then ultimately I can click the run button, which will then spin up the computers and then begin the analysis. And so I'll show you in my other demo project, I have an output of one. And so this is a very simple BAM tools index. So I ran BAM tools index. This was over a year ago now. And so this was obviously a very simple one where I ran the alignment. I fed in an alignment, a BAM file, and I needed to generate an index for something. And so it was very rapid. It only took, let's see, I believe probably about a couple minutes, 5 minutes, and then the price was 3 cents for this. And so what I like about the tasks is that each of these are saved. And so, here I am a year and a half later, and I can go back and track that BAM tools run that I did, or the alignment that I did, or the variant calls that I did. And all the settings are saved. Everything is preserved here in this space. And so it's very easy to go back and say, what did I do? What exact setting did I set on the caller? And so it's all preserved here, which makes reproducibility very, very easy for running this type of analysis. And then if I want to do this again, then I can click this blue button, and then I can plug in a new BAM file and run it as well. So then from this point, once I have, if you have that long list of genes, your differentially expressed genes maybe, and you want to run it through your R script or do a Python analysis, then you can feed that over into the interactive analysis tab, which allows you to be able to, again, run the data cruncher to be able to generate your graphs, generate your tables, and really look at the data up close and explore it in more detail. So I'll end my tour there, but know that that's available. So you can either download those outputs to your computer, or you can continue to work here on Chromatica as well. All right. So that is my tour. And so hopefully from that point, you can see how you might start all the way on the Kids First portal. And again, explore the different data sets that we have, and then take those all the way to the point of where you're running the analysis and generating the figures for a particular study. So we've got about 19 more minutes, and I don't think there's any more questions in the chat, but if there are, please fill them in, because we're going to have time for questions here at the end. And so while you are thinking about different questions, I might show off some of our different Kids First resources that can help you either get started, or if you're stuck looking for more assistance, this can be a great place, a great first place to view. All right, so on the Kids First dashboard, so this was that first page that I landed on. You'll notice up at the top here, so we looked at our different tools, but then up in the top right, there's a resources tab. If you click on that, it'll provide a drop down, and that resources. So this is a great place for getting additional help and getting more information about the different Kids First data sets and Kids First resources. So the first one page I'll show you is this support page. And so if you click that support link, it will take you over here to our Kids First DRC Help Center. So these are our training and support documents that we have provided for users who use the Kids First DRC and our data sets. If you scroll down, it takes you down a little bit below. So everything from getting started to our FAQ pages. But specifically for today, I'm going to highlight the studies section here in the middle. So this is our studies page. And so if you click on studies and access, it opens up this great page where it shows each of those different Kids First studies that we have available. And it provides all types of information about the different studies that can link you out to get you started. So here's the top first 8 of our studies. Again, we're up to about 27 studies at this point on the Kids First portal. But if I click on the congenital heart defect study right here, it brings up its own card, which gives us more information. So this is one of our oldest studies that came out in 2018. It gives us information about where the files were sequenced. And then it has these 2 useful links here. And so those 2 useful links, the top one, which has the PI's name, that links you out to the study abstract. And so that will send you back to the Kids First program website, which gives you more information about when the study was funded, how the study was designed, and has the words by the, written by the PI themselves while they were providing the study. The second link, which has the PHS number, will take you directly to the dbGaP page. So all the Kids First data is controlled through dbGaP. And so if you're interested in using Kids First data sets, you can submit an application on dbGaP in order to be able to receive access. And so if you're interested in this particular study, you can click this one link and it will take you directly to dbGaP, where you can then click that request access button, and that's the first step to being able to work on these files yourself. I also want to highlight, so we have a lot of training that's available. And so our partners at the NIH Common Fund data ecosystem have provided a great set of resources for training as well. So they have a full set of tutorials that walk you through first creating an account to being able to move files over, and the different features of the cohort builder as well. They just created a recent one, which is very good, about doing an RNA-seq analysis. And so it goes from identifying the files on the portal all the way to running a complete analysis and then going into the data cruncher and generating the graph. So it's a really awesome tutorial, and it's a great resource that can help folks get started and then plug in your own data, but follow those same directions as well. So our friends at the Common Fund Data Ecosystem have provided a really great resource of training that's available. And so this is just a screenshot of that it shows, this is the different button, click here to learn this. It's got some great pictures. We also host a monthly office hour session. So on the second Tuesday of every month at 3 o'clock in the afternoon Eastern time, you can join me as well as members of our data operations team and our bioinformatics team, and we are all available. It's an open office hour, so anybody can come and bring questions to the table, and we answer everything from how do I get started, how do I find files, how do I apply to dbGaP, all the way through I did this analysis and this is my result, what do you think of it, and then everything in between. So it really is an open office hours, and we encourage everybody to join. The link is there. We also post it on our social media page as well. One of our newest features is the Kids First portal, Kids First forum. And so we use this to post announcements. So we recently, for example, updated our thematic workflow. And so we posted a blog post here, and as well as users can provide questions to our forum as well. So the Kids First forum is a relatively new resource that we're looking to expand out. And then, of course, you can always reach us at our support inbox. So we are support@kidsfirstdrc.org. Please reach out to us. Me and about half a dozen other people check that inbox every day. And so if you send an email to that inbox, we'll find it, and we'll get back in touch with you to try to help you and support your Kids First research. And so at this point, if we were in person, I'd love to hand you some conference goodies like a pen or a notepad, but unfortunately we can't do that, through the virtual medium. But I would love to share with you a virtual kind of take home. And so that's these support slides here, which I can share into the chat, I believe, if I can share it with everyone. That right there. So that tiny URL will take you to the page that has these two slides on it. So this is your virtual take-home gift. And so here what we're looking at is we've got the Kids First DRC support resources. And basically, this is a lot of that same information that I provided throughout the talk, just condensed into 2 different slides. And so we have all of our information about the email inbox, the different homepage, the different help centers and training materials. As well as for our advanced users, we have links to the GitHub and Kavatica links to our different Kids First pipelines, as well as some directions about the Kavatica API if you're looking at operating from the command line. All right, and so that is everything that I had prepared today. And so we still have about 12 minutes, and so I'm available to answer any additional questions that folks might have. So please feel free to ask any additional questions. Otherwise, you can, as I said, you can always reach out to us, support@kidsfirstdrc.org. So that's our support inbox, and that's what we use for all of our Kids First DRC communications. We can use that to continue the conversation after the meeting. And we please encourage you to stay connected through our social media accounts. So that's where we post everything from updates to our pipelines, announcements about Kids First webinars, releases of new data sets, and then events like this, our office hours, and our events like here at ASGH 2021. And so you can find us on social media and Twitter. We're at Kids First DRC, and we'd love to connect with you, and we'd love to again support your research. So with that, thank you again so much for joining. And if you have any further questions, then I'll be happy to take with them. Otherwise, I hope you enjoy the rest of ASGH 2021. And we'll see you around the conference. All right, I see a couple more questions popping in here, so I'll run down the list as they've been coming in. So Owen asks, will the recording be available? Yes, so we are going to record this session today, and we're going to post it on our Kids First YouTube channel. And so then we'll be able to make that available to everybody, and I'm sure we'll probably post it on our social media accounts as well. Kids First DRC will be where we share that. The next question is asking about, can you explain the variant tab on the main Kids First page? Sure. So the Kids First page, if we go over to Kids First, so variant is one of our new features, and it's still developing. So I skipped over it a little bit, but the variant database is a way to be able to search Kids First data sets for particular variants of interest. And so you can search on the variant level, you can search on the gene level, and it basically searches the Kids First data to be able to find, are there particular participants that have mutations in that particular gene. And so if we look, for example, if we type in, maybe my favorite gene is BRAF, and so I want to learn and see what are there variants in BRAF in our particular Kids First data sets. So I can click BRAF. And then what it does is it provides a list of variants that were identified, and then it has any particular associated consequences with those variants or mutations, as well as any studies that those have been identified in. And so then we can click a little bit further. So if we, for example, this one seems to be relatively common, then I can click on this particular mutation, and we can see that it's found in 4 studies. We have within 143 participants. And so from here, it gives us some more information as well. It tells us more about BRAF. It gives us some different, so these are the different studies. So it highlights the 4 different studies that it popped up in, as well as the different participants. Then you use some external information, so external cohorts as well. As well as any clinical information associated with either that particular gene and other, as far as different ontologies and different data sets that might be available. And so from here, again, so at this point we have these different studies. So we found it in these different data sets. And so basically what we want this tool to be is that this can allow you to then be able to identify which Kids First studies to apply to. So if you know that it's applying in Kids First CDH, this is the diaphragmatic hernia study, then it will pull this up. Perhaps I can apply for these 3 studies on my dbGaP application. So the variant tool is very, very cool. We're very excited about it. Unfortunately, right now, it's still in kind of the developmental beta test type phase. And so we only have 6 of our 27 Kids First studies in here. So it's still, doesn't represent the full set of Kids First data, but we're hoping to finalize that by the end of the year and have a larger set of studies so that it is more valuable. So that's the variant tab. So it is very powerful, but we've only got 6 studies in here, so that's why I didn't include it in the main talk today, but that's a great question. And then the last question was, do you have any data on retinoblastoma? And so I don't believe there's a Kids First retinoblastoma study, but we can, so I can show you, for example, we can look it up right here. So if we go on the Explore Data tool, and I start a new page here, and I can type in my search box retinoblastoma, and then we can see, do we have, it seems that we have 6 patients that were diagnosed, have the retinoblastoma mondo diagnosis. And so then within that associated, we can see there's 1 study. So within the Kids First Intersections of Cancer and Structural Birth Defects study, yes, 6 participants were diagnosed with retinoblastoma. And so, so we don't have a full retinoblastoma study with hundreds of participants, but in this broad study, which looks at multiple cancer types and multiple structural birth defects, it does seem that, yes, there is some data about retinoblastoma. So that's how you would do that search. All right. Well, I'm not seeing any more questions, but thanks so much again for everybody who joined. Of course, I want to thank all of my partners here at the Kids First DRC. So I'm the person who gets to stand in front and share our wonderful website, but there's certainly a huge team of folks from our computer engineers to our data analysts to our bioinformaticians and everybody who makes the DRC run. And so I definitely want to acknowledge all of their hard work. I especially want to acknowledge the work of our great communications team who helped me put together this presentation and helped me operate our webinar here today. So thanks so much for joining us this afternoon. Again, I hope you enjoy the rest of ASGH 2021. And please feel free to reach out if you have any more questions about using the Kids First DRC's data sets or platforms. We'll be happy to support your research. Thanks so much. Have a good afternoon.